Good morning. I am Shubhangi Rohatgi, PGT Biology, KV number 1, AFS Hindon, and I welcome you all to the third and final session of Chapter 6, Anatomy of Flowering Plants. In the last class, we had studied in detail the epidermal tissue system and its components like epidermal cells, the stomata and the epidermal appendages, that is the trichomes or hairs. We had also studied how the opening and closing of stomata is regulated by the help of guard cells. Then we had studied in detail the anatomy of dicot and monocot roots, and we had learned to compare the two also. We had seen that there is not much difference between the anatomy of the two, except for a few points like the monocot root is polyarch with 6 or more than 6 vascular bundles here, and the pith is well developed as compared to dicot root where 2 to 4 vascular bundles are present only. Also, a major difference was that monocot roots do not show any secondary growth. Why? Because cambium is absent there. Secondary growth is shown only by the dicot roots. Then we studied the dicot stem. This was the slide we had studied. It is a typical dicot stem showing the epidermis, the cortex, hypodermis, followed by the pericycle and a well developed pith. We also saw that the vascular bundles are arranged in a ring and they are open vascular bundles because they have cambium present between the phloem and the xylem. When we compare this structure with the monocot stem, we found that the pith is absent, but the ground tissue is present, which is largely parenchymatous, and the vascular bundles, they are enclosed by a bundle sheath layer and they are scattered all over the stem TS. Lastly, we had studied the structures of dicot leaves and monocot leaves. We saw that in dicot leaves there are two different types of epidermises, the adaxial one and the abaxial. Also, there are two different kind of mesophyll cells, the palisade and the spongy mesophyll, as compared to the monocot leaf where only one type of epidermis and one type of mesophylls are present. Because of these, the monocot leaf is also known as isobilateral leaf, and the vascular bundles, they are arranged all through the length of the leaf. Now, this was about the last class. Let us move on towards the today's topic, that is the secondary growth in dicot plants. We know that primary growth means increase in height. Here you can see that primary growth is resulting in the increase of the height of the plant. It results by the action of apical meristem and intercalary meristem also in monocots. So, primary growth is mainly concerned with only increase in height. New cells are being formed at the tips and they are being used to increase the height of the plant. On the other hand, secondary growth occurs by the activity of secondary meristematic tissues and it results in radial growth, that is the increase in girth of the plant. So, while primary growth is concerned with increase in height, the secondary growth is concerned with increase in girth of the plant. It occurs after the part of the organ has completely developed. This is again a very important point. While the primary growth is concerned with the growth and development of the organ as it on its whole, secondary growth is concerned with the later maturation. So, it will occur only when the part has completed its growth, and once the secondary growth has started, primary growth won't take place in that particular region or that particular part of the plant. Lastly, the secondary growth does not occur in monocots. So, let us see this figure here. This is LS of a plant showing different regions. Here is the meristematic tissue where new cells are being continuously formed, and here they are being elongated and matured elsewhere in the plant's body. So, here the process of primary growth is taking place, where new cells are being used to increase the height of this portion of the plant. However, as we move to the older portions, we can see that the increase in height has already taken place. Now, the secondary xylem and secondary phloem have started to form, and because of formation of these new tissues, we find that the region here is more in girth as compared to here. That means, the secondary growth has resulted in increase in the girth of the plant. Moving on. So, secondary growth occurs by the activity of lateral meristem, and this lateral meristem can be of two types, vascular cambium and the cork cambium. Again, vascular cambium is of two types, intrafascicular cambium and interfascicular cambium. Look here in this diagram, the cambium that is present between the xylem here in the red patches and the phloem is known as intrafascicular cambium, and the one which develops later in the medullary rays between two consecutive vascular bundles is known as interfascicular cambium. So, vascular cambium is of two types, intrafascicular cambium and interfascicular cambium. What happens? What are the steps of secondary growth in dicot stem? First of all, a complete cambial ring is formed. Initially, cambium is present only in a single layer in vascular bundles between xylem and phloem only. That is what I just said. This cambium is known as intrafascicular cambium. Later, what happens when secondary growth is to begin? Cells of medullary rays adjoining the intrafascicular cambium become meristematic and form interfascicular cambium. Due to this, a complete cambial ring is formed. If we are to draw, we can draw it like this. Suppose this is the part of the stem where secondary growth is to begin. Now, initially the vascular bundles were arranged in such a manner that there was phloem and xylem and in between these two we had the intrafascicular cambium. Many such bundles were present inside before the beginning of the secondary growth. What happens when the secondary growth is to begin? The first step is development of interfascicular cambium in the medullary rays region of the stem. Here the cells were parenchymatous in nature and now they have de-differentiated to become meristematic again. So, this green ring is the interfascicular cambium and the red one which was present earlier is the intrafascicular cambium, intra and inter. What happens next? Well, this cambial ring that has been formed will start dividing. The vascular cambium that is present now in the plant has two types of initials, the ray initials and the fusiform initials. The ray initials are the one, the derivatives of which will produce the rays, and these rays are horizontally oriented. However, the fusiform initials are those cells whose derivatives will give rise to secondary xylem and secondary phloem. So, vascular bundle works for both the purposes. It is going to eventually give rise to rays as well as the xylem and the phloem also. Again, how does a cambial cell divide? When a cambial cell divides, it produces two daughter cells. One of those daughter cells undergoes differentiation to produce a xylem or phloem mother cell as the case may be. And this xylem or phloem mother cell will later divide or differentiate to form xylem or phloem elements. However, the other daughter cell, it will retain its meristematic activity and continue to be a cambial cell. Why? Why is this division of fate? It is because the cambium has to continue to work and continue to produce xylem and phloem for a large part of life of the plant. It is not so that it divides only once and a lot of xylem and phloem are produced and that is it. No, the process continues for quite a long time, and that is how the cambial cell perpetuates. What it does, only one of the daughter cell is used to produce xylem or phloem, while the other is used to perpetuate the cambial ring also. This way both the cells are present there. Now, the cambial cells divide tangentially and secondary xylem and secondary phloem are added on the internal and external sides respectively. The cambial cells continue to divide in this fashion, producing secondary tissues on either sides. Now, what is happening here? Let us first learn to draw the cambium. Whenever we want to draw the cambium, we should draw rectangular, radially flattened cells. This is how we draw cambium. And if you remember, in the last class, we had discussed how to draw phloem. Whenever we draw phloem, we try to draw two types of cells. Like this. One type of the cells will have nuclei present. Can you tell me which are these cells which are rectangular in shape and have nuclei present? Yes, these are the companion cells associated with the sieve tubes. And as far as xylem is concerned, you must be remembering that in order to draw xylem, we draw vessels with thickenings. These cells are not completely round, somewhat polygonal. So, this is how we draw xylem. So, I have drawn a portion of a vascular bundle with primary phloem, cambium and primary xylem. I want to raise a question before putting on the labeling. Now, as I have said that the cambium will start dividing and it will start producing phloem on the outside and xylem on the inside. My question is that where will the secondary phloem and secondary xylem be placed with respect to the primary xylem and primary phloem? Will the order be secondary phloem, primary phloem, cambium, secondary xylem and primary xylem, or it will be like secondary phloem, primary phloem, secondary xylem and primary xylem? Which of the two options is correct? Yes, you are right if you are thinking that this one is correct. The source of the new cells is the cambium. So, the new cells will be produced near the cambium and the older tissue which was present earlier will be pushed towards the cortex if it is phloem and towards the pith if it is xylem. So, after the cambium has started functioning, the secondary phloem will be formed near the cambium on the outer side, and secondary xylem will be present near the cambium towards the inside. In this way, the xylem, primary xylem and primary phloem, which were near in the beginning, they will move apart as the cambial activity continues to take place and more secondary tissue is formed. So, here is a diagram which is also showing that near the fusiform initials of the vascular cambium, we find the secondary xylem and secondary phloem. Afterwards, there are primary xylem and primary phloem. Now, what happens? As you move towards the pith for xylem, you will find the older elements. The younger will be near the cambium. Similarly, as you move towards the cortex, in case of phloem, you will find the older elements. The younger will be near the cambium. So, generally it happens that cambium produces more xylem than the phloem, and cambium ring with all the tissue in the front of it is pushed more and more towards the periphery. A lot of xylem is being formed, and what happens as the secondary xylem is being made, the cambium is pushed towards the cortex because a lot of tissue is being made in front of it. So, it is forced to move towards the cortex. On the other hand, the amount of phloem that is being produced is not that much. The primary xylem and primary phloem, which were lying side by side in the primary condition, are gradually pushed apart from each other. This is how the stem would look like after a certain amount of secondary growth has taken place. What is happening? These red patches represent the primary xylem. It has moved towards the pith and a lot of secondary tissue has been formed here. And this is the secondary phloem, and outer part is the cortex. So, you can now see that the increase in girth has already taken place. Talking about the characteristics of secondary xylem elements are more or less similar to those occurring in primary xylem. Vessels or tracheae, whichever may the case be, are most abundant here, and they are usually shorter and much larger than those of the primary xylem and are mostly of pitted types. That means they bear the piths. What about secondary phloem? Well, secondary phloem elements are produced on the outer side of the cambium. As we just saw, they are produced on the outer side of the cambium. Usually, the number of phloem elements produced is less than that of the xylem elements. In most of the dicots, the primary phloem gets crushed with increase in thickness, and secondary phloem takes over the physiological functions and it remains active for a considerable length of time. What happens that because of so much tissue being formed inside the stele and the cambium is moving backwards and more of secondary phloem is also being formed, the primary phloem gets crushed in the process. Meanwhile, the secondary phloem that has been formed, it will now take over the functions of primary phloem and do all the work, that is the conduction of the food. And lastly, the formation of medullary rays. If you remember, initially we talked that there are two types of initials in the vascular cambium, the fusiform initials which give rise to the xylem and phloem mother cells, and the ray initials, the derivatives of which will produce the ray cells. What are rays? The rays are the parenchymatous tissues which are present between the vascular bundles. When these rays are present in the primary stem, they are known as primary rays, and when they are resulted in after the secondary growth, they are known as the secondary rays. So, ray initials of the cambium ring form some narrow bands of the parenchymatous cells. These cells extend radially from the pith to the phloem. These are secondary medullary rays. The rays present in the xylem are xylem rays and the rays present in the phloem are the phloem rays. So, what will happen? Let us draw a portion of the stem again. Suppose this is the cambial ring which is producing the secondary xylem on the outside, secondary phloem on the outside and a lot of secondary xylem on the inside. Meanwhile, the ray initials, what will they do? They will start producing the parenchymatous cells instead of the elements, that is the xylem or the phloem. Such rays will be present in the xylem as well as in the phloem. So, what will happen after some time? We will find bands of parenchyma present in between the xylem and the phloem also. Now, I am drawing it in lines. These bands of parenchyma are nothing but the rays. So, the ones which are present between the phloem will be the phloem rays, and those which are present between the xylem will be the xylem rays. This was about the activity of the cambium in the secondary growth in dicot stems. Let us perform an activity to understand this phase, these steps of growth in a different and a better manner. I will now use different colored papers to show different parts of a stem, dicot stem. Suppose this is the initial structure where this ring represents the cambial ring. The blue portions are, you will now tell me the names. If the situation is like this, the brown patches are xylem, primary xylem, and the red patches are showing or representing the primary phloem. So, initially when the secondary growth has just begun, the stem will look something like this. We will not show the other tissues, the parenchyma that is present everywhere, which is the filler tissue, or even the cortex for that matter, because right now we are just concentrating on the mechanism of secondary growth. So, we are just making the vascular bundles and the cambium. So, this is how a stem would appear in the beginning of the secondary growth, when the secondary growth has just started. I was to ask you, what do these blue patches represent? And what do these orange patches represent? If this is primary phloem and this is primary xylem, what do these represent? Yes, you are right. These are intrafascicular cambium and these are interfascicular cambium. And this is how a stem would look like in TS when the secondary growth has just started, when the cambial ring has just formed. Now, what will happen when the later portion of the secondary growth resumes? The cambium, the fusiform initials, they will start cutting off new cells quite quickly towards both the sides, inside for secondary xylem and outside for the secondary phloem. Let us see, if the secondary growth has begun, what will happen? The phloem, primary phloem will be pushed apart further away deeper into the cortex. Similarly, the primary xylem, it will also be pushed inside towards the pith, something like this, and you can see that in the process some of the xylem and phloem are crushed, and the spaces that have been now formed, these are not the spaces, actually the phloem and xylem, they have been pushed because of the formation of secondary tissue. Let us show the secondary tissue with a different color of paper. So, I am representing the secondary xylem with yellow colored ring and secondary phloem with this green colored ring. Okay. So, our rangoli of dicot stem TS is almost ready. Now, something is missing. Yes, the medullary rays. The ray initials which are present here in the cambium, they will start cutting the parenchymatous cells to produce the medullary rays. This is it. So, can we try to label the different layers that we have drawn here? This was primary phloem, it has moved deeper into the cortex and the place has been taken by the secondary phloem. Inside to it was the cambial ring and further inside we will see the presence of secondary xylem followed by the primary phloem, and of course, in between there are medullary rays. This is how the secondary growth in stems takes place. You can see that the girth has increased of the plant at that portion where secondary growth has occurred. Let us compare the two pictures or drawings that we had just drawn. So, here let us try to label them. As we had just discussed, the blue portions represented the interfascicular cambium and the orange portions represented the intrafascicular cambium. The brown paper was used to show primary xylem and the red ones showed primary phloem. The places in between the vascular bundles, it was the medullary ray. It is a time when the secondary growth has just started and a complete cambial ring has formed. Afterwards, when the secondary growth has occurred, we see that some secondary phloem has been formed and because of this the primary phloem has been pushed outside. The cambial ring is still there and it is followed by the secondary xylem which has been newly formed, and the primary xylem is also there, but it has been pushed towards inside. Further, we see the development of medullary rays by the action of ray initials which cut off parenchymatous cells inside and outside as well. So, when the rays are present in the phloem, they are known as phloem rays, and when they are present in the xylem, they are known as xylem rays. This was about the secondary growth in the stele. You can compare the structure of the two. This was just at the beginning of the secondary growth, and this is after the secondary growth has completed. You can see the girth has increased, you can see a lot of new tissue has been formed, a lot of wood has been formed. Now, what happens? Imagine a situation when you are carrying a paper bag and you keep on putting in stuff in it. You are stuffing it with so many things. What will happen? It will eventually tear. The same thing is going to happen to the plant also. Meanwhile, in the cortex, nothing is happening, but inside the stele, a lot of new tissue is being formed, and the cambium is pushing towards the epidermis, towards the cortex, because of formation of so much of secondary xylem in front of it, and even at the back of the cambium, a lot of secondary phloem is also being formed. So, what is happening? The girth of the plant is increasing, but it is increasing from the center. The center is expanding, and if the peripheral parts do not expand in the rate similar to that of expansion of the center, what will happen? The peripheral parts, they will rupture and the plant will die. To avoid this situation, what happens? Secondary growth takes place in the cortex also, and this is the formation of cork cambium. Secondary growth, wherever we talk of secondary growth, we will talk about cambium. When it is the vascular cambium, the secondary growth will take place in the xylem and the phloem. When it is cork cambium, the secondary growth will take place in the cortical area of the plant, that is the outside the stele. So, what is happening? Formation of secondary tissues in the intrastellar region exerts pressure on the epidermis and other extrastellar tissue. The epidermis ultimately tends to rupture. Why? Because of so much tissue that has been formed inside. Some of the parenchyma cells in the cortex now become meristematic. Again, how will the parenchyma cells become meristematic? By dedifferentiation. These cells now represent the cork cambium or the phellogen. The cork cambium starts exhibiting mitotic activity on both the sides. So, now it will start cutting new cells on both sides, towards inside and towards outside also. The structure of this cork cambium. Phellogen or the cork cambium is much simpler than cambium. Why? Because it consists of only one type of initials. I will repeat that in vascular cambium, we saw that there are two types of initials, ray initials and fusiform initials. Ray initials give rise to the rays and fusiform initials give rise to xylem and phloem. However, in the phellogen, there are only one type of initials which produce both the sides, both the layers. These cells are rectangular and radially flattened. They are compactly arranged without intercellular spaces, and they have vacuolated protoplast also. So, again let us try to draw this phellogen. Whenever we draw any kind of cambial cells, we will remember that the cells will be rectangular and flattened. So, when I am drawing the cambial cells, I will draw straight lines. A few layers, suppose I want to draw, and then I will make these cells rectangular. The nuclei can be shown because meristematic cells have always a conspicuous nucleus which is very active because it is dividing. Now, let us say that this is the cork cambium. By the way, are you clear where this layer has developed? inside the stem. I will draw a quick small diagram for you. You can see it here now. Suppose this is the stem and this is the stele where lot of secondary growth has taken place now. A lot of xylem has been formed, a lot of phloem has been formed, and this area is the cortex, which is now about to rupture because of development of so much secondary tissue. The cork cambium, it will develop somewhere here below the epidermis in the cortical region. Okay. Now, when I say that towards outside, I mean towards the epidermis, and when I say towards inside, I mean towards the cortex. So, the cork cambium that has been formed below the epidermis in the cortical region will now give rise to two types of cells, the cork on the outside and the secondary cortex on the inside. That is what is written here, that the cork cambium cells, they divide tangentially producing a phloem, that is the cork on the outer side, and phelloderm on the inside. Phelloderm is also known as the secondary cortex. Let us see this figure. It will make more clear the arrangement of different layers inside the stem of a dicot plant at this point of time when phellogen has developed and it has started cutting off different layers. So, let us assume that this portion is the stele where secondary growth has taken place and is still taking place. So, the phellogen or the cork cambium, it will develop somewhere here. This yellow band, this represents the phellogen or the cork cambium here, and it has developed below the epidermis in the cortex. Now, when the phellogen will start cutting off new cells, it will form a layer of cork or phloem towards outside, that is towards the periphery, and a layer of secondary cortex or phelloderm towards the inside. So, these are the layers that will be visible in a stem TS at this point of time. Talking about the properties of the phloem or the cork, much more phloem is produced on the outer side than phelloderm on the inside. You remember the vascular cambium, it worked just the opposite. It produced more of xylem towards inside and lesser phloem towards outside. On the other hand, this phellogen, that is the cork cambium, is working in an opposite manner. It is producing more phloem on the outside and less secondary cortex towards inside. The cells of the phloem, that is the cork cells, are compactly arranged and they have no intercellular spaces. They lose protoplast after differentiation and become dead. Their cells remain filled with air and colored organic matters. Also, a thick layer of suberin is deposited next to the primary walls. So, what are cork cells basically like? They are colored, they are suberized, they are thick walled and they are dead. They will not allow any movement of air or water through them. Let us try to draw some cork cells. As we had discussed in earlier classes also, this is how we draw thick walled cells without intercellular spaces. I will draw in this manner, increasing in height. When you are drawing the cellular diagrams, it is very important that you try to differentiate between different types of cells with your way of drawing them. It is important. Otherwise, your diagram will hold no meaning to a layman's eyes. This is how we draw thickenings. And you know what? We use colors also in the paper, not to make our diagrams aesthetically appealing and beautiful. No, we use colors because we are using them for coding our diagrams. That brown color here represents cork and the pink color here represents the cork cambium, so on and so forth. So, it is better that you always try to use different colors or different ways to label your diagram while you are making it. So, I will draw a few cells here only in order to save some time. This is how the cork cells would look like. They are colored, they are thick walled and they are suberized and dead also. Cork is also known as the phloem. The cork cambium is producing lot of cork cells towards the epidermis, towards the outer side of the stem. On the inner side, however, it is producing the secondary cortex. Can you see this color? Okay. Secondary cortex cell are similar to the primary cortex cells only. So, they are parenchymatous, they have intercellular spaces, they are polygonal, isodiametric, have protoplast, just like the primary cortex. So, can you really see different types of cells formed in one diagram? This is how we draw the diagram to show different types of cells, not only labeling them with different names. It will not work. You should try to draw the cells according to their nature, and with practice you can learn it. So, this is the secondary cortex. I hope this color is visible. As I had just said, moving back to the cork cells become impervious to water and gases because of the presence of layer of suberin. The phelloderm, that is the secondary cortex. The phellogen produces phelloderm on the inside towards the cortex. The cells resemble those of the cortex. These are living, isodiametric and with intercellular spaces. This is what we have drawn. You can see the intercellular spaces here. The intercellular spaces, these are visible and they are isodiametric, living. How can you show that I have made these cells living and not these? Yes, the presence of protoplast that I had drawn with the dots shows them to be living. However, presence of very thick walls and lumen and absence of nucleus shows them to be non-living or dead. These cells, these secondary cortex cells may carry on photosynthesis and serve in storage of food also. What is periderm? These three together, taken together, the phloem, the phellogen and the phelloderm, these three make the periderm. The cork cambium and the secondary cortex, they together form the periderm. What is the function of the periderm? Hold these three layers. They protect against desiccation and mechanical injuries. And why are they able to give protection? Because of strong suberization of the cork cells and their compact arrangement. Because of this, they can protect against desiccation and mechanical injuries also. Now, what is bark? we have been talking about the structure of the stem for such a long time, and we have not for even once mentioned bark. What is bark? Well, the bark is the name given to all the layers that are generally present outside of the phloem. The periderm formed at the superficial region of the organ may not be adequate to withstand the pressure coming from inside. A lot of secondary growth is taking place here. Additional layers of periderm are formed in the deeper region of cortex, pericycle and even phloem. Now, whenever periderm will be formed, Koch cells will also be produced. So, Koch cells are produced on the outer side, and the living cells present on the outside of the phalogen, they stop getting supply of water and food because the Koch cells are suberized. Now, all of them will become dead, and these dead tissues taken together, they form the bark, or as I had said in the earlier simpler manner, that all the tissues that are present outside of the phloem constitute the bark. Can you differentiate between periderm and the bark? So, the periderm is made up of only 3 layers, the coch cambium and the cortex. On the other hand, bark includes all the tissue layers right from the epidermis up till the phloem. All the layers will be included in the bark, and bark generally slows off. So, this is the picture of the bark that is given in your NCERT book, and this is the diagram showing the bark. You can see after bark they have drawn the phloem. So, all the tissue exterior to phloem makes up the bark. Lenticels. What is happening? The stem is now increasing in the girth, the bark is being clawed off, the epidermis, it gets ruptured, and a lot of suberized cells have been formed which are impervious to water and air, especially air. So, how will the inner tissues get gaseous exchange? Lenticels help in this function. So, the periderm has replaced the epidermis in protective function, and dead Koch cells with suberized wall are impervious to gases, and they have been formed in the cortex. Gaseous interchange has thus become difficult. Now, some pores which look like lens shaped raised spots on the surface of the stem now develop and take up the function of gaseous exchange. These pores are known as the lenticels. Lenticels are mostly formed just beneath the stomata. What happens? The parenchyma cells near the substomatal chamber, they lose chlorophyll and divide in various planes, forming a mass of colorless loose cells called as complementary cells. These complementary cells give rise to lenticels. The diagram given in your NCERT book is very clear. So, you can see here that the Koch cambium was there. You can note the arrangement of cells, they are rectangular and flat. So, the Koch cambium was there, but somewhere in between complementary cells have been formed. These complementary cells are very loosely arranged, they are parenchymatous and they have raised a bit, and because of this, because of the formation of lot of complementary cells here, the epidermis gets ruptured and a lens shaped opening is formed here. This opening is known as lenticels, and it will allow for the gaseous exchange. Otherwise, wherever the Koch is present, the gases will not be able to be exchanged. Now, what do you see in this picture? This is a typical log of wood. You must have seen such pictures or logs of wood anywhere. Yes, beautiful concentric rings, and you must be knowing that these are annual rings. How are they formed? Usually, in the plants growing in tropical regions, cambium is active throughout the year, but in temperate regions, cambial activity may cease with the onset of unfavorable period and enter into a dormant state till favorable conditions are available. And because of this seasonal activity of the cambium, distinct growth layers are formed in the xylem, and these growth layers appear as rings in the TS. Since one layer is formed every year, so these rings are also known as annual rings. Formation of annual rings is characteristic in plants growing in temperate climates with pronounced seasonal variations. They are not so much seen in plants growing in tropical regions. What is early wood? Well, the wood that is formed in the spring, which is most favorable season for plant growth, is less dense and is made of vessels with wide diameter. When you need to conduct lot of water, so the vessels will be formed with wide diameter, and this wood which is formed in spring where active growth is taking place. So, lot of water is being used and larger diameter vessels are being formed is known as the early wood. So, the late wood, it will be just the opposite. When the growth is not happening at such fast rates, the vessels will be formed that will be narrower and dense. This wood will be known as the late wood, and yes, it will be developed in the autumn. Why? Because water requirement is less in the winters. So, because of the presence of early wood alternating with the late wood, we see the formation of rings in the plant. Here you can see in the early wood there are large vessels, however, in the late wood the vessels are very small, and because of these alternate patterns we can see the formation of rings. Sometimes false annual rings also appear because sometimes the cambial activity can be disturbed by defoliation or because of some disease or adverse climatic conditions like drought also. In that case, the development of wood is checked and growth is resumed later in the season, resulting in development of false annual rings. Have you heard these terms, heartwood and sapwood? When a lot of secondary xylem has been formed, we can see that there are two woods, the heartwood and the sapwood. The sapwood is the one which is lighter in color. It is also known as alburnum. Here the living cells are present in addition to the tertiary elements and fibers. That means the parenchymatous cells, xylem parenchyma is also present here. So, what does it do? It carries on the physiological functions, primarily conduction of water and solutes and support and storage of the food also. If that was the sapwood, what will be the heartwood? Yes, the darker portion. It is also known as the duramen, and it mainly consists of the dead elements. It is dark colored and it is the hard central part, and when it is hard, what is it going to do? It is going to provide the mechanical strength. Further, it also carries gums, resins, oils and tannins. Then the function will be to provide strength, and commercially it is more strong, durable and compact. So, it is more valuable than the sapwood. How sapwood transforms into the heartwood? The cells of the sapwood, they lose the protoplast, their water and food contents are lost and tyloses are formed. Tyloses are balloon like outgrowths that occur through the pit and they obstruct the lumen. This is a tylose. The walls of parenchyma cells also undergo lignification to make the heartwood, and finally, a lot of secondary metabolites are also stored inside the heartwood. How does secondary growth take place in roots? The process is similar, just the difference is that vascular cambium originates from the many tissues like below the phloem bundles, pericycle tissues and even above the protoxylem. And initially it is wavy in nature, later it becomes circular. Later process of the secondary growth is same, that the cambium will cut off xylem towards inside and phloem towards the outside, and this is how secondary growth will take place. Now, what are the advantages of heartwood, can you think? Yes, it carries a lot of secondary metabolites which can have antimicrobial properties, and the cell walls are lignified to provide strength. This is an interesting question here. This fence has somehow incorporated into the tree while it was growing. So, Now, if the top of this fence is currently 2 meters above the ground and the tree grows 0.5 meter every year, how high will be the top of the fence in 3 years? What will happen? Will it take it above? No. Actually, the top of the fence will be only 2 meter above the ground. Why? Because I had told you initially, if the secondary growth has started, the primary growth will not take place now. So, the stem will remain at this point only. The height of the tree is increasing, but from the tips only. Here the height is not increasing. Another question. This is the stacking cone analogy, and that is how we explain this concept of secondary growth. Suppose one cone is added every year because of secondary growth. So, initially the height was this, then another cone is added. The height has increased and at the base only the girth has increased. This is how 2 cones means 2 layers, 2 years, 3 cones means 3 years and 4 means 4. Now, at the A point the stem is 4 years old here, and here at B point the stem is only 2 years old. Okay? This is the difference between primary growth and secondary growth. The primary growth is taking place here and secondary growth is here. One question, where would you want to core a tree to determine its true age? Obviously, near the base. Why? Because the older portions are there. You can be asked similar questions in the exam to identify the regions. We had discussed that this must be the bark, the phloem, the cambium, the xylem and the darker colored heartwood. And questions like to differentiate between heartwood and sapwood can also be asked. You can mention that they are the differences there in the color, the weight, living and dead cells and functions, and also the tylosis. Well, this was about the chapter 6. I hope that you have enjoyed studying this chapter. Thank you very much.